All right, can you guys hear me?
Yes.
Yay! All right, everybody. We are ready to go here. Hello and welcome everyone to Part 3 Clinical Trials 101. My name is Casey Craig. You may know me from our SCNAA Family Support Face Group page as Stella's mom or the SCNAA Family Book Club lady. But today I'm serving as the Acute Syndrome Foundation volunteer and will be moderating all of the clinical trial readiness education series. This 5-part series is aimed at helping all of us gain perspective and information as we prepare for the upcoming clinical trials. The Acute Syndrome Foundation wants to ensure that all of our families are informed on how clinical trials work, as well as general drug development, ethics of patient data collection, and potential future therapies. This series will bring together panels of industry professionals, clinician researchers, and advocates to discuss these important details. Last week, we had the pleasure of chatting with 3 amazing panelists regarding patient data. They discussed the importance that we know about the value of our data and our rights. One of the most profound moments of that discussion is when Dr. Gabby Belfort made a comment about the fact we should always ask the question, what are we getting out of any study that we participate in? It was most definitely an aha moment for me. If you missed part 1 or part 2, no worries. We are working on uploading a recorded session for you to view at your convenience, so stay tuned. Now moving on to today's event, Clinical Trials 101. In this discussion, we're going to cover the basics of clinical trials, how they are designed, what kinds of considerations go into trials for very rare diseases. As always, this session is going to be an informal discussion, so please ask questions as we go. Before we get started, let's have a couple of housekeeping things. Number 1, group interaction through questions or comments, or if you just need something to be repeated, please feel free to do that. And you can do that with the chat box. So to your right, you're going to notice a chat box, and it will allow you to type in those questions or comments or anything that you need from Hillary or our awesome panelists today. To your left, you'll see that there is a tab called Expo, and this area is where you're going to find some troubleshooting kind of pinpoints for you. But if you're also having issues and you can't find Expo, or if it's not answering your question, then we have our fabulous technical guru, Megan Varner, on the other end, where you can't see her, but she's doing all this awesome work and she can help you tackle that. So just go straight to the comment box or the chat box and ask for help, and Megan will help you. All right, before we get started, I want to take a moment to give a very warm welcome to our Duke 15 community that are joining us as we partner together in this clinical readiness journey. I also want to extend a huge thank you to our 2 sponsors for this event, Praxis and Zogenix. Now I will hand over the torch to our very own Hillary, let's see if I can say this right today, Savoy. We are here, all know her as director and founder of the Acute Syndrome Foundation, but more importantly, she's the mom to the original Cute, Esme. She's truly paved the way for our kids, and I know I'm forever grateful for her leadership and her friendship. Miss Hillary.
Hi, everyone. Sorry, it does take a second to switch over. Thank you, Casey, and well done on the Savoy. I am so happy to be here again with all of you, with our SCNAA families, and again, pleased to welcome the Duke 15Q community. As Casey said, I'm the founder and director of the Acute Syndrome Foundation, and we're here again today for our 3rd session in the 5-part series on clinical trial readiness. And today's topic is Clinical Trials 101. In some ways, it sort of seems like maybe we should have started with this as our first topic, Clinical Trials 101, but I think we really set the stage for this discussion today with our 2 previous sessions, focusing first on the meeting with Rachel Callender and her Awesomeness Forum, where we talked about what we as caregivers already know about our children and what's already perfect about them. And to my mind, this is kind of where we start from, where we should focus on as we hope to move towards clinical trials and build on what is already amazing about our children. In our second session, as Casey said, we learned about the value and uses of patient data I also had flagged Gabby's statement about what we get out of any sharing of our data. And I think it was a really profound moment. And when we share our data, it can be used to do all sorts of things. And one such example is that it can be used to directly develop plans for clinical trials. And that is something that we can get out of participating in sharing our data. For instance, your child might not be in a trial, but you get to help in the creation of a trial for a community. So for instance, that's really amazing. When we did the Cute Syndrome Foundation and Xenon survey this time last year, and the results of which are circulating now in the support group, thanks to Neurocrin putting together some slides for us. This is something that we all participated in, sharing our data, something that we can do to help our community. And just to be clear about what participation in something like that looks like, you would expect to see the name of the pharma company, official IRB approved language, and all that good stuff that we talked about last week. But how is that data helpful? How does it translate into endpoints and inclusion and exclusion criteria? Or really, what even are endpoints and exclusion and inclusion criteria? And we're going to talk about that today. I'm sorry, I have a cold, so pardon my coughing. There is always kind of a great deal of excitement around the idea of clinical trials and engagement with the pharmaceutical industry, but we also need to firmly grasp sort of general notions about how clinical trials work, who will be in them, and how all of that is determined in order to be fully informed participants, which is, of course, our goal. I have the good fortune of being in the inside of a number of these kinds of conversations with our pharmaceutical partners, helping to explain our community, helping to design protocols and advocate for our needs. And I get to learn a lot from really exceptional leaders in this space. And I want all of you to have that opportunity as well. Hence our amazing panelists here today. I'm going to welcome to join me on the screen. Oh, there's Luke. Might take a moment. Oh, okay, Carolyn. All right. And we did have some technical difficulties earlier, so we'll give John a moment to join. But in the meantime, I'll go ahead and introduce Luke Rosen, who is the VP of Accelerated Development and Community Engagement at Ovid and the founder of KIF1A.org, and also the father of a lovely little girl with KIF1A. And Carolyn McMicken, who is the Director of Medical Science Liaison at Neurocrin Biosciences. And hopefully soon we'll have Dr. John Millichap joining us. He's Director and Attending Physician of Neurology and at Children's Hospital. And I'm afraid we might have lost Luke there too. Carolyn, can you hear me? Oh. Good, okay. Oh, and here's John, yay, we all made it.
Can you hear me okay?
Yes, yes, absolutely, wonderful. So I am gonna ask all of you to introduce yourselves in a moment, but I just wanna make a few notes that, as always, these panel discussions are super casual conversations. And families, if you have questions along the way, we're here for you. We're here to talk about the things you want to talk about. So feel free to use the chat box to ask your questions along the way. But also just a note that, there may be details of current programs that our panelists can't answer questions on. So we just want to be mindful of that, but ask your questions and we will answer them to the extent possible. So starting with you, John, do you guys each want to take a turn at introducing yourselves and giving just a little bit of background about what brings you here?
Sure. I'm John Millichap. I'm a pediatric epileptologist here at the Lurie Children's Hospital in Chicago. So I've been working in epilepsy for over a decade, taking care of children with all types of epilepsies, but I became very involved with genetic epilepsies in about 2012, and I've continued to expand my understanding and my involvement with different parent groups and over those years, and become involved in several clinical trials, which has been really exciting as more and more pharmaceutical companies are getting involved and interested in specific genetic causes of epilepsy. So really 10 years ago, none of this was happening, and today it's becoming more and more common. Very exciting.
Some of the families may be having a little bit of a hard time hearing you, John. So I don't know if you can bring the mic a little closer.
Can you hear me now?
Yeah. Oh, that's great. Thank you.
Should I repeat what I said? I don't know.
I think we got most of it, but just going forward. Thank you.
I'll hold it up like this. Sorry about that.
Wonderful. Thanks. And Carolyn.
I'll do a mic check first. Can everybody hear me? Well, thank you so much for inviting me to join this discussion today. As Hillary mentioned, I'm Carolyn McMicken, and I am a medical science liaison director with Neurocrine Biosciences. And in fact, a discussion maybe for another day is the role of what medical affairs does. But part of the reason I come to join this panel today is that in prior roles in pharmaceutical industry, I've had quite a bit of interaction with the rare epilepsy community, supporting programs for Lennox-Gastaut syndrome, as well as infantile spasms and refractory partial seizures. And I've just always been so inspired by the epilepsy community, and in particular, the parents and caregivers of kids that have rare epilepsies. I also, prior to being in pharmaceutical, worked in psychiatric clinical trials in a clinic, so have that perspective of how they run, and we worked with kids as well. And I do sometimes see that there is an information gap for families, and really wanted to participate in helping dispel some myths and answer questions, hopefully in a language that resonates with you, and be as transparent as possible.
Thank you so much, Carolyn. And Luke.
Hey there. Can you guys hear me now? I think I follow suit and do a sound check. Yeah, awesome. Thanks, John, Carolyn, and Hillary. Thanks for having me here, and for all of the community members who have joined. I'm sure that I know many of you, and it's hard not to see you in person, as we often do around this time of year. So thanks for tuning in. Like Hillary said, I'm Luke Rosen, and we were talking on a sort of panel prep call yesterday, and I have a lot of hats, but I've found that they're effective if I just wear one hat. That's really bring my perspective as a father to a kid who has a very very rare genetic epilepsy. I didn't have, much to do with science at all, or certainly not biotech, certainly nothing to do with clinical trials, until in 2016, when my then 2-year-old daughter, Susanna, was diagnosed with a mutation in her KIF1A gene. And we knew of maybe 25 other folks in the world at the time. And that's comes along with different characteristics of developmental epileptic encephalopathies, like a lot of us are affected by these families here. And my wife and I started a foundation, KIF1A.org. And the goal of our foundation was really to support, and is still to support, research leading to treatments. And there was a part of that journey where we were really developing this robust, strong preclinical program with academic collaborators and others. And it was the time where it was, and then what? Like, what do we do now? We've done things like created a registry, found more people, advocated for genetic testing, and are really, but people started saying, it's time to partner with biotech. That's the next step. And I didn't know what biotech was. If you asked me, I probably would have said a robot or something. And then, I went back to school to study a little bit about what everybody was talking about so I could just keep up with the conversation. I studied specifically bioethics because I knew that we were going to have access problems. If the average timeline from discovery to commercialization or market, and Carolyn and John, you might know this better than I do, but 12 to 14 years at one point was what I heard. And I knew that our kids didn't have that much time. So my goal was to tighten that window. And so I Googled rare neurology biotech in New York. I live in Harlem, and Ovid Therapeutics popped up. So that's when I met the folks at Ovid, and I lead what we call the Ovid Accelerator, which is just what we're doing now, and that's pulling in all of our collaborators and families and foundations, and making sure that our process is there isn't that lag time of a few years in between finding out that our kids have these disorders and working with incredible physician advocates like John and advocate industry members and incredible folks like Carolyn. If we're all working together faster, we can just be more efficient with research and development. So I lead the Ovid Accelerator, accelerated development, and inherently in that role is community engagement with our rare disease community and specifically our rare epilepsy community. At Ovid, we have two programs. One is rare neurodevelopmental, and one is rare epilepsy, or DEEs, and so much of that is crossover. So primarily my work is with the research and early development teams, because nothing can happen if we don't bring the community and the families and the foundations in as early as possible, so we can develop things like the appropriate outcome measures and endpoints, and design clinical trials that are powered for rapid development. That's me.
Thank you, Luke.
Pleasure to be here.
Thanks, all three of you for joining today. I really, I'm always so happy I get to have these conversations with all of you and with our community. So I think with this, I think we just really start at baseline. So why do we have clinical trials for potential therapeutics? I think, John, maybe as the clinician here, you can help us understand what clinical trials are and why we need them to get from an idea to treatment for our kids.
Sure. Can everybody hear me now?
It's a little quiet.
Can everybody hear me? Can everybody hear me now?
That's better. Yeah.
Is it okay now? Okay. Yeah. So sorry, my desktop didn't work, so I'm on my phone, and I hope this works okay. So yes, that's a great question. I think at a very high level, you would think about all the medicines that are at your pharmacy for whatever reason. Everything that you take is regulated. Whether it's over the counter or from your pharmacy, whether it's for high blood pressure or kidney disease or your kid's stomach ache or your kid's ear infection. All of these drugs have gone through this process. Most of them, unless they're very old, have gone through this process to make sure that they're both safe and effective. And I know there's a lot of talk about clinical trials and bringing medications quickly to the population with regards to our current pandemic. So I think maybe some of these words are becoming, very common and more understood by the general population. But when we think about epilepsy drugs, they go through a process for many years where we have models in small animals typically, or in other now computerized models, where we screen compounds that may result in a decrease in electrical activity in the brain or help to control seizures. And they will screen thousands and thousands of drugs, and they'll whittle them down to a few that seem to possibly help control seizures. And from those small numbers of drugs that might help control seizures, then they whittle them down to ones that don't cause intolerable side effects. as best as they can tell. And it's those few candidates that get through to, further studies in other animals or other types of clinical studies. And then very few of those will then get to humans. And, typically the first stage will be testing these compounds in healthy adults who volunteer for such types of trials to make sure that taking the medicine at a dose that would, potentially help the disease it's designed to help would, not cause too many side effects, or to get an idea of how the body metabolizes it. So that's sort of a general overview of how a chemical or a drug might come to clinical trials. And I think it's important to realize that, some of these chemicals will come from nature, come from plants or trees. Aspirin is from a tree, but some of them are formulated in the lab and created through organic chemistry. Everybody thinks back to high school and chemistry class, and so I'm mixing different chemicals together. We can create reactions and create new compounds or new chemicals. So really these are chemicals, and then they become drugs once they are tested in humans and make it to your local pharmacy. So we really need trials to determine why what drugs are going to be helpful for patients with epilepsy, and which ones are safe to take. And we'll find out sometimes after a drug has gone through all of these steps and has been tested for many years and released to the market that there is some additional side effect. That's happened several times in my experience. So we're constantly learning and monitoring these drugs that we prescribe to children to try to help their condition.
Wonderful, thank you. And Carolyn, I think it would be really helpful. John mentioned, the pandemic kind of making us more familiar with some of these processes, which I think is really interesting. But obviously, we're talking about an incredibly rare population, and so I imagine the process for designing a clinical trial in a rare population looks a little different.
It does. So the main similarity is that for whether it's a rare population or a condition that's affecting many millions of people, that the same ethical and safety standards apply. So we don't want to compromise that, regardless of how anxious we are to get a new treatment to market. And so it involves a lot of careful planning for whatever you're studying. One of the main differences with a rare disease is that it would be a rare by definition. So you have to be even more careful in getting it right, because you're not going to have as many chances to go back and gather more information with that limited population. So there are processes in place that kind of help move drug development along for rare populations with those considerations. So the FDA, will grant, say, a rare disease designation or orphan drug, and depending on the quality of data that you can collect early on, there may be fewer studies required to get to an approval. But there's still a lot of those steps in understanding the condition that go in front of that, which is another challenge, right? Because some of rare diseases like SCN8A, in the grand scheme of things, have only very recently been identified. And so we don't know as much about some of these rare conditions as we do about something like high blood pressure. So there's still a process of trying to understand as much as we can to get the best trial design.
And that's actually, I think you bridge perfectly for me, Luke, as someone who's bridging that role of advocacy and biotech. Can you talk a little bit about how, as a patient community, we can help make sure that we're setting ourselves up to be as helpful as possible to to the biotech world when they're trying to design trials around us.
Yeah, thanks, Hillary. That's such a great point. And I would almost say that, clinical trials 101, before that comes clinical trial readiness 101, right? And I feel like that's something that the community can always be doing, but you can't do it without, partnering and communicating with our, the medical community and the biotech community. And Carolyn's point is such a good one, and that's that before you have to understand the disease so well in order to design a clinical trial that's going to be impactful, and by understanding the disease, what I mean, and certainly John can speak to this better than I can about these our genetic disorders that we're just sort of starting to understand are so heterogeneous in nature that think about, and now I can speak just as a father of a kid with this disease, and knowing other families in the disease, is that we all have different challenges, right? It's not all seizure reduction. There has to be other meaningful outcome measures and endpoints that can change the course of the disease. And I used this example yesterday. It would be fantastic to have a therapeutic that could allow my daughter to stand up and run a marathon, right? But it would be life-changing if she could have 5 more steps, right? So it's what really matters to the family. So back to your question, Hillary, is if we as a patient and caregiver community are able to be just clear in our organization and articulation and our communication about what matters most to us and what a meaningful change in our lives actually looks like, then I feel as if the medical and the biotech and the research community can kind of aggregate that importance of change and what what would make a big difference in our lives, and turn that into something that's actually targetable and capturable. And you can't fix it if you can't measure it and capture it. So I think that it becomes about how to capture what's incredibly meaningful to the families and what a therapeutic change would look like.
That's perfect. You use the term endpoint and outcome measure. So I think right there on my list, you guys are just knocking them down. I'm really impressed. Thank you. My next thing on my list was, let's talk about nomenclature. So Luke, what's an endpoint? What's an outcome measure? Are they the same thing? What are we talking about here?
Right. So there are different, in clinical trial, there are primary endpoints and then secondary endpoints, and they're basically targets of what we're trying to accomplish in a clinical trial. And the most frequent endpoint and again, John and Carolyn can speak to this way better than I can, as I am sort of new to this, is that we think about seizure frequency reduction, right? And in an epilepsy clinical study, and that's because it's very capturable and very something you can clearly see. So an example of an endpoint in an epilepsy clinical trial is seizure reduction. It's a capturable and sort of very you can see signal and you can see change clearly with an endpoint in a study, and that's really what defines whether or not the study is successful or not successful. Is that accurate, Carolyn and John? I'm sure there's a clearer, more
John,
better description.
Yeah, Carolyn, I'll just comment there and then pass to you. I think, Luke, that's a really good point, and I think that the seizure endpoint works really well for more general trials where we're looking at adults, say, who have certain types of seizures, focal seizures or generalized seizures, and just in general from whatever cause. But the issue we're talking about today and the one that is on my mind more is these specific causes of, you mentioned DEE, which stands for developmental epileptic encephalopathy. Those are children who have seizures, but then it's so much more than that. It's their sleep, their development, their behavior, other issues that are also important to quality of life. And I more and more, as the former administration started this precision medicine initiative nationally. Really, what that means for epilepsy is finding a treatment that might specifically target the underlying cause or the underlying etiology of the development and seizures. And so I think that the medical community and the pharmaceutical industry and FDA is, it seems to be moving away from just relying on seizures for approval and looking more at measuring improvement in life. And I kind of look at some of our adult problems that receive a lot of funding and attention, like Alzheimer's disease and Parkinson's disease. Many of you have probably heard of those trials. They're measuring cognitive improvement, how people stand up and walk across the room. These are these are measurements that are used to approve drugs to treat those diseases. And some of the similar things, like as Luke said, walking 5 steps is amazing. And for a Parkinson's patient, that's also amazing. And they're getting drugs approved when it makes that happen. So I think that there's precedent, and we just have to start thinking about our patients and these rare conditions in a different way.
You got
I
I don't know
think
if you could see, John, but from Casey, yes, 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 amen on your
Actually,
comments
I
about
cannot
these.
see chat on the phone.
Well, so you got a yes, 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 amen on your comments about the various things that our families and our children are dealing with. So well done. And I think this actually, Carolyn, maybe I could ask you to speak to inclusion and exclusion criteria. I think that's another thing that our families are used to hearing discussed, but maybe we haven't necessarily been walked through what they mean and how that applies to a clinical trial.
Sure. So inclusion exclusion criteria are very important when conducting a clinical trial, and it's really, again, everything is about balancing safety, minimizing risks, and maximizing our chance at detecting a benefit of a treatment. So typically the inclusion criteria are there to define who we're trying to help and study. So it's typically you need to have this condition and diagnosis, and all of the things you need to have in order to participate in the study. And when you get to the exclusion criteria, these are the things that, again, we want to minimize harm. So these are the things that may not make somebody an ideal participant in a clinical trial. So they might relate to coexisting medical conditions. It might relate to, what other treatments you're currently taking. And that could be, again, for a variety of safety reasons. It might relate to whether the person has somebody who's available to take them to the study appointments. So again, it's there to protect the patient community, as well as to ensure that the data. You hear sometimes people say that you want to get clean data, and that's in the earlier stages of clinical trials, it is important to get clean data so you can have as clear information and answer as possible in order to move to the next step.
And I think that leads me to the term open label extension. Speaking of next steps, could you explain what that is? Because I think, again, it's a term that gets tossed around a lot, and I think sometimes it isn't fully explained to us.
Can I
Sure.
just follow
Go
on
ahead.
something
Yeah.
Carolyn
Yeah.
said without interrupting, because it's such a good point about, and it loops back to what we were saying earlier, is that when thinking about inclusion and exclusion criteria, you can't really do it right or correctly or accurately if you don't really know the community and the disease, right? So I think that it harkens back to that idea of community engagement and Hillary, clinical trial, the community can do to actually enable the folks who are designing those clinical trials to make the inclusion and exclusion criteria appropriate for the disease. And by that, I mean, I'll use my daughter as another example in our disease, is that we know that Susanna pretty much is my daughter. She plateaus on most of her AEDs or anti-epileptic drugs. Usually it's like 4 months is around the time where we end up having to change drugs, right? So but if you look on, and a great resource and the sort of default resource for clinical trials is clinicaltrials.gov, which we can pop in the chat or something. That's the sort of database where you can just punch in a disease and see what clinical trials are happening. And that's also where you can see the inclusion and exclusion criteria for each trial. And I saw a trial that had, you have to be stable on X amount of AEDs or anti-epileptic drugs for X amount of time, right? And my daughter wouldn't, and nobody in our community would be included in that trial, because the folks who, while the drug might be effective, the folks who were designing the trial didn't take the time or didn't know that, well, our kids are never on between X amount of AEDs and X amount of AEDs for X amount of time, because they all plateau after 3 months. So there's no stability, right? So if you know the community and learn with the community and the clinicians about those specific diseases and the nuances of those diseases, you can really design a clinical trial that is appropriate for that disease. Is that, I don't know if that's clear, but, and then so critical is an open label extension. So back over to you, Carolyn.
Well, and I wanna piggyback on what you just said, Luke, because I think part of our discussion yesterday was a little bit about how that's actually a changing picture, right? As in a very rare community, when I joined the SCN8A Facebook support group, we were 30 people, and now we're over 360 something. And so the clinical picture has changed as the community has grown, and also as the criteria for testing has changed, right? So different groups of individuals are being tested. And so we're finding different people. Like, for instance, we have we have a phenotype that is non-epileptic, but they're not typically being tested. And so we don't know how big that population is. So I think what you mentioned yesterday was a little bit of a discussion about how you account for that in engaging the community and collecting data about the community, so that you're getting a snapshot that's good for a period of time, and then maybe needing a snapshot later down the line.
Yeah, and I think it's colleagues like our foundation colleagues and John, like Dr. Berg, who, if you look at there's a recent publication that the LGS Foundation leaders and the Dravet Foundation leaders and members of the research community published together, and it was called Non-Seizure Consequences of DEE. And so I think it was a 2-year study with over 200 families, and it was looking at exactly that, right? Like, what are the different domains or the different areas of disease that, if we are able to create or identify a therapeutic that would address those different domains of disease, it would be meaningful. So those collaborations and articulating them in a way that is scientific and published is so important. So.
Carolyn, I do want to come back to you to explain to us what an open label extension is. I love it when we digress, but I want to be sure we get this one down.
Well, although I could circle back to more on the inclusion exclusion. Is it okay if I do that?
Yeah.
So
Yeah, absolutely.
just the one kind of, again, comment regarding what Luke mentioned is that in your standard clinical trial that's not with a rare a DEE epilepsy group of individuals, there is, well, in anything for safety reasons, you want to ensure that the individuals you enroll into a trial are quote unquote medically stable. And obviously that's a huge challenge for a lot of families of children with epilepsy with DEE. And I'm sure Dr. Millichap, John, could comment on that, and being a clinician, and when you're thinking about who might be appropriate, looking at inclusion exclusion criteria, how you think about that and communicate with families. It would probably be helpful for the group to hear.
Sure. So the patients who are having, the most number of seizures, you would think would be, probably the best patients to be in the trial, because they might see the most benefit. But if they are having so many seizures and their primary doctor sees fit that they, change medication very frequently, or they're being admitted to the hospital with other issues, illnesses, or having so many seizures that they are admitted to the hospital frequently, then they may not be the best candidate for a clinical trial, because the information that we would obtain from that individual being in the trial may not be helpful for measuring the endpoint, and therefore the purpose of the trial, which is to get good information that would help from the most common patients who are suffering from the condition, and not necessarily the patients who are at the outliers of the bell curve. So those who are having maybe very few seizures, where they're not having enough to be qualified for the clinical trial for a seizure primary endpoint, or having too many seizures to qualify for the clinical trial that's testing seizures as a primary endpoint. But with rare diseases, I think we might be more inclusive, or sometimes we discuss that we want to be. I think Caroline said we have one shot, or Hillary, that we really want to select the right patients to answer the question, so that we can get the drug available if it is helpful to the community at large. And then in there are other avenues for those who maybe either have fewer seizures than are needed to be eligible for the trial, and those who are having too many can then maybe get the drug once it is FDA approved, or in other open label type studies that are looking at other features like safety. Does that kind of answer it?
And that's a good segue back to the to the open label extension.
I invited the right people. Well done.
Yes. So open label extensions most frequently are used to continue to assess the safety of a medication. So if you have a study that somebody's considering participation in, occasionally they will have a potential for what they call rollover. So you finish the double-blind, randomized, placebo-controlled trial, you have the opportunity to roll over, meaning continue into this open-label study. And what that means is that you complete this one trial period, you still don't know in that initial phase if your child was on an actual experimental intervention, medication, or on a placebo. But in the open label extension, you will get the investigational medication and continue to be followed up for safety, and also still measuring the efficacy, though a different kind of standard applies in the eyes of the FDA in terms of measuring efficacy, because they really want to see that placebo control. So open label is really about that safety, and you still can get some long-term efficacy data as well.
I just came up with a question I've not had in the past as you're talking, that if you were in the clinical trial and you were on placebo, which you would not know, when you roll over to the open label and start on the drug, how do you handle a titration? Not because as a family you might notice. So how does that work?
So there's different
protocol.
ways of doing it. Yeah, it's again often what they call blinded. So sometimes in the initial phase of the trial, everybody will be tapered off of the medication and then, or placebo, and then start back. So that's kind of how you handle that. You wouldn't necessarily expect to continue on the same dose level as you were in the original trial, and that there's more flexibility in an open label extension where if other medication changes need to be made, often then they're allowed in that phase of the study. So it's more, it mirrors real life treatment more than the initial phase.
Okay.
Important to note also is that that one of the most vital players in all of this is certainly the study team and the clinician who's running the study, and safety always being paramount in everything we do. But there also needs to, people should know, and I don't think I knew this before I became involved in clinical study, is that not every single phase 1 clinical study or phase 2 clinical study has a placebo arm, right? So often an open label extension, and that's to say that there are clinical trials happening that don't have a placebo, and you always need a baseline to see change against, right? Now, excuse me, whether or not that baseline it could be very strong natural history study. It could be, it varies from case to case, but an open label extension also happens with studies that don't necessarily have a placebo arm. So, but it's important to know that there are different protocols and different studies. So when you go into sign up for clinical trial, and that process is something we should talk about from when you consult your physician to how you find out about a clinical trial to the next step, and then to the next step, right? They're very specific stages of enrolling in a clinical trial. And I think that when you are, just like everything, informed consent is so important. And, there needs to be very specific understanding of, it's actually 2 different studies, and you always have the option to roll over into an open label extension. And, I think one of the greatest benefits of an open label extension is that if there is something that is efficacious and safe, and there is something that a family is feeling benefit from, it's not that a study ends, that however many long week clinical trial ends, and then all of a sudden it's, what now? All of our kids with seizures have withdrawals, if we stop taking anything right away. And so there's an opportunity that if something is impacting in a positive way, that you don't have to stop and wait for commercialization, right? And so that's a major component of an open label extension as well.
And I think that's actually a really good chance to move over to a question that we have here, because we've established that not everybody can be in a clinical trial, right? And there's good reason for that, whether it's their stability, their seizure frequency. So to you, John, Casey asks, what do you say to families if their children don't qualify for clinical trials? She imagines there might be some disappointment.
Sure. So I'll discuss with every family and clinic if there are any clinical trials that are publicly announced that their child might be affected by, and certainly screen for the inclusion and exclusion criteria very formally, and if the family either doesn't want to participate after they hear the details, that sometimes happens. The family may be interested to be part of a clinical trial, but once they hear about the requirements to be in the study, the number of visits, any blood draws or other tests that might need to be done, they may decide that they actually don't want to participate. That's one situation. But if they do want to participate, but they don't meet the inclusion and exclusion criteria, I will, counsel them as I think you already mentioned several times, Hillary, that they have opportunities to contribute to the further development of treatments and our understanding of their child's disease by contributing information to observational studies that are not interventional studies, which have different inclusion and exclusion criteria. So I would say for SCN8A, there's lots of studies to be involved in. Just not every individual child will be eligible for every interventional study, and that's really for their safety and also for the to get the action that the FDA to make a very important decision.
Thank you. And we have another question from Jesse and Rob McNabb. How do we find out if our children do qualify, especially if we're living outside the US? I think, Carolyn, that might be a good question for you.
Sure. We already put the clinicaltrials.gov link in the chat there, and if you find a specific trial and scroll down on the page, it usually lists, while not necessarily the specific sites, but it will list kind of the locations in the US as well as other countries that there are sites in, and there's typically some kind of central contact that you can then call to get more specific information on where those sites are exactly located. And sometimes, you might start a trial program with a certain number of sites, and then you come to find out that there is a number of people looking for participation that are in a different part of the world or a different country, and then kind of go back and evaluate whether or not to open up another site in order to facilitate that enrollment. So it's a complex process.
I think that's a really good point, and something that is an actionable item that foundations, families, and the community can help facilitate, so that we can kind of cut through that unnecessary step of having to stop and reevaluate and see where, right? So if we're able to put together something as simple as a heat map about our community, right? I'll use just my own frame of reference, our KIF1A community, right? We have a heat map where we put together, we know here are all of the places that would be we call it in biotech and in industry is called feasibility studies, right? And what the feasibility part of all of this is, is quite literally, setting up, picking the clinics and setting up where it's going to be most, where we're going to be able to include more and enroll more families, right? And so if the foundations are able to say, look, we have a lot of families here in this region and here in this region and here in this region, and the sponsor, or a sponsor is just another word for the company that's running the study, can pull in and work with the foundations early. And we do this all the time at Ovid, right? Is that we work with the foundations to get a list of where the families are and what clinics they go to, and that's part of feasibility that is a way that families and foundations and communities can accelerate the whole process is by coming to the table with that and saying, you know what, this is easier. Here's where everyone is. You know.
And we do have that in the 8A community. The Acute Syndrome Foundation has essentially a heat map. And we have one in the US and a global heat map as well that sort of allows us to track where we know patients are. And I think it's also just fair to say that if there is a clinical trial happening in this community, you know, you will hear about it in the support group. We will make sure as a foundation that you know everything that we are allowed to share as soon as we are allowed to share it. And that's part of my job, is to essentially advocate to have as much information as possible out in the community as early as possible.
And that has to include the phone number or the email of somebody at the company, right?
Yeah.
I mean, if the company can't be, you know, if there's a question about inclusion and exclusion criteria, obviously always the first person to go to and to talk to is your physician. Always. That's a no-brainer. Or it's, you know, always the physician. But, you know, if there's a question about inclusion and exclusion criteria, it shouldn't be a third party who you get sort of directed to answer the question. To, you know, there's always needs to be a phone number or an email address with somebody who is part of designing that clinical trial, who can answer the question of, you know, well, I live in the EU and this doesn't look like a global clinical trial. How can I have my family consider participation, right? It's always about having the information to decide whether or not this clinical trial is right, appropriate, safe for your family. And, you know, the only way to really get those answers is to speak with somebody at the company who's sponsoring the study and the clinicians who are helping to run the study. So I think a big part of it is communication and who you can talk to. I mean, don't be afraid to just call every single company that has a neuroscience department and say, is there a clinical trial? And are there plans for a clinical trial? And where are they? You know, I mean, it's half my day is spent, you know, talking to families about just that. And if they don't meet inclusion exclusion criteria, the first question has to be, well, why didn't they meet inclusion and exclusion criteria? And is it a flaw in our study design? And if the answer is no, it's not, then that is a really difficult conversation to say, you know, the study was designed this way, and that's why the inclusion exclusion criteria is such. And unfortunately, it doesn't seem that that this study is right for you. But also, then the company has to go back to the clinical development team and say, I don't know if this quite makes sense. Do we need to file what's called an amendment? And what an amendment is the ability to change a protocol in a very safe and in a very, you know, regulated way. But it's kind of like calling an audible in football, right? It's knowing and understanding that this might need a little bit of tweaking and not stopping the study, but staying the course. And I think that often inclusion and exclusion criteria fall under that category of needing an amendment or needing to be reevaluated.
I want to be sure we have two more questions that I know, and be mindful of everybody's time, but I want to be sure that we answer these questions because I think they're both really important. So all questions are important, but these are particularly. So Carolyn, of course, you're going to get the question that you knew you would get. So Alex asks, on the topic of clinical trials, do we have any kind of timeline for the SCN8A kiddos at this point? And I want to be sure to be clear for everyone that Carolyn is with Neurocrine, and they are, you know, have been working with our community on a potential clinical trial that comes from a licensed program that came from Xenon, who all of you are familiar with. And they are one of the companies in the space. And I'll let Carolyn say what she can say about that.
Sure, and I imagine, but Hillary knows better than I do, that the community saw Neurocrine's press release last week that we did hear back from the FDA, and they've requested additional non-clinical data before we proceed with the phase 2 clinical trial. And you know, what we can say now is that both Neurocrine and Xenon are working to address the FDA's questions, and our goal is to initiate that trial next year. I know that this probably isn't as much information as we'd all like to know. I think, you know, especially right at this moment in time, there's just uncertainty everywhere, and it would be nice to have better answers. That's what we can say right now, and we are dedicated to addressing the FDA's question and moving forward with the program. We did get the rare pediatric disease designation, so we think that's a very positive sign that we're on the right track in trying to pursue a treatment for SCN8A.
And I can also add that I'm working very closely with everyone at Neurocrine and making sure that, our community knows everything that they can know as soon as possible, and that Neurocrine is aware of, our community and our needs and all of that. So those lines of communication are open, as they always have been with all of our partners in the space. And so, we will bring you information as soon as we possibly can. And then the last question from Tammy, I think this is a great thing. It didn't come up in our sort of nomenclature conversation, but Tammy asks about, for children who don't qualify for studies, but they've tried everything, and if they're in a serious situation from a health perspective, what is, if any, what processes are possible for accessing an emergency access route? And I guess, John, do you want to speak to that? So usually that's clinician initiated, is that correct?
Sure. Yes. So there's several situations where we've done this actually commonly for certain conditions where the most common situation is actually a drug is in use and approved in Europe or other countries besides the United States, and therefore produced and available commercially, just not in the U.S. And in those situations, we would file a sort of individual FDA approval. We call it an IND. So it'd be the FDA and the physician writing a very detailed report to the FDA why this individual patient, in their opinion, should be allowed access to this drug that is not currently available at the general pharmacy, stating that there's safety information from other agencies in the world that can be referred to, and experience with this particular drug in that population. So I think that's the most common situation where access to an unapproved drug is granted. And then the second is compounds which haven't been through the testing that we've been referring to, the phase 1, the phase 2 trials in the US or anywhere else, really new, novel compounds which may be effective for that person's specific condition, and writing an IND to get access, emergency access to that drug for very specific reasons. So I think that's less commonly done and less commonly successful, but it is possible. But I would say in sort of the more rare situations, and it's ideal to put efforts into the clinical trial for a group of patients that we might be able to have more generalizable information collected and move that potential compound further along the line where it might be available to all, rather than exposing one child. It's a matter of exposing one child. We would be exposing a child to the risk of the medication or the compound, but not necessarily wall as far as getting potential FDA approval. Some
It's
rare instances, this
also important to know that every sponsor or every company, the FDA requires that every company has a statement about what their early access program is on their website. So you can always find that on the company's website. For instance, if you go to Ovid's company website, there's a section that says our early access program is, or we don't have an early access program, or here's how you go about applying for early access. So there's always information about how to go about that with each, or again, at the end of the day, just reach out. And yeah.
I think it's important to note. Oh, go ahead, John. Sorry.
Yeah, that's a good point, Luke. Well, I was just going to say that's a good point. The physician and the family might be interested in this, but if the compound is not
And
available,
I think it's important to note that for
certainly a
emergency
roadblock.
access, you would need, as a family member, you would need a willing advocate clinician to make the ask. And then, as Luke says, you need to have a willing company on the other side to be able to answer that ask. And any compound that might be considered for something like an early access would have to have been tested to a certain point. So it would have had to have gone through a phase 1 trial,
or it could be that there's just the timing was not right? So the trial ended enrollment and isn't going into their next phase until X amount of time. So you're in that window where the family would be, the physician who's initiating the request believes that the family would benefit from this, but there's just no trial to enroll in right now. But there is, but there's an open label extension. There is a study that had happened. So to me, that's a time where it's appropriate to make that request. I mean, it always is if your physician believes that it is, necessary. But I think a big part of it is also communication, nomenclature, lexicon, right? So what's the difference between compassionate use, early access programs, pre-commercial access? And, there's so many different ways of thinking about it. But at the end of the day, it's all the same, right? And it's about just asking, and if the company and the clinicians can work together to clearly articulate the pathway forward and how to go about it.
There's also N of 1 studies that those aren't 101. It's true. Yes.
Well, but at the end of the day, it's how do I get the
those
drug?
aren't
How do I get into the
101,
trial? That's pretty 101.
Yeah.
Yeah.
And I think that is, from a parent perspective, that really is the big question, is when can I help my child and how can I help my child? And that balance, and part of why we've done this series is because we have to balance that sense of hope and wanting to help our individual children, but also wanting to help the whole community, and having realistic expectations about timelines. As Luke said, it's not that long ago that we would be told 10 to 12 years before anything would move. And here we are seeing things move significantly faster. I remember talking to JP about the compound that Neurocrine now has licensed, and him saying this has moved faster than I ever thought possible. And so, this is really an amazing space that we're in, and an amazing time that we're in. And on that note, we've gone way over time. I'm sorry, unsurprisingly, because this is such an important conversation, and maybe we need a follow-up that's a little bit beyond 101. But I do want to wrap up in consideration of everyone's time, and just say thank you, Luke, Carolyn, and John, for your willingness to come on and talk with us today, and share in this really important conversation. Luke has put his email in the chat, and I can speak directly to, he means it, email him.
Yeah, reach out anytime.
He's great.
It's most likely that I won't have the answer, but I can certainly put you in touch with the person who does have the answer.
Yeah, and thank you, Casey, for the intro, and Megan for the tech support, as always. And thank you to the community for showing up, and a big thank you to our sponsors for today's event, our Praxis and Zogenix. And we will see you guys next week for a conversation about setbacks and failures, which I think will be really, really interesting. Luke wants to join.
But I don't want to be a panelist answering questions.
It's going to be a great panel.
Yeah,
It's going to be a
I know,
really
I'm
great
sure,
panel. We'll
sure.
have
Incredible.
Bernard from Praxis, Gail from Zergenics, and JP from Xenon. So look forward to seeing everyone here next week. And thank you again, panelists.
Thank you.
Thanks,
Have a
Hillary.
great day.
Bye, everyone.
Thanks, guys.